I'm going to start off with an introduction of what pheochromocytomas and paragangliomas are. And I looked up the phonetic pronunciation because I thought that might be useful, because I think if you're diagnosed with one of these, I think the first thing to get your head around is actually pronouncing what you have. But when I looked up the phonetic pronunciation there, I don't think that actually makes it any less intimidating. I think that actually, if anything, makes it look more intimidating. But I thought I would just keep it there anyway, just to I think emphasize how challenging these tumors can be, even just trying to pronounce the name of them. So what are these? Pheochromocytomas are tumors that arise in the adrenal glands, specifically arising from this inner part of the adrenal gland, which is called the adrenal medulla. And paragangliomas are tumors that come from the sympathetic nervous system, which is all the way down from starting at the base of our head, the bottom of our head, and go all the way down to our bladder. So paragangliomas can pop up at multiple parts of our bodies. These are very closely related tumors, which is why we always talk about them together, because the cells that they come from are very similar. They're not exactly the same, but close enough that we can use the same treatments on them. And some of the things that start off these tumors are the same between the two different types. So what are these? What is that adrenal gland and that sympathetic and parasympathetic nervous system? What is that all about? Well, it's all about the fight and flight response that we all need for survival. So when we've got something scary coming up, like a presentation, then our fight and flight response kicks in, and that's our cells from the adrenal glands and those cells from the nervous system starting to produce this hormones, adrenaline and noradrenaline. And we've all had that experience of what it feels like to have adrenaline and noradrenaline going around our body. Our heart rate gets faster. We often hear the sort of pounding in our chest and pounding in our ears. Our muscles get more tense. We start breathing more quickly. Our body diverts blood away from our digestive system to the parts of the body that need to be ready to go to fight off whatever predator, or in a modern day, anything that's frightening us. Just a little aside that it also has effects on the liver, that gets more glucose going around our body as well, so that our body is really primed to suddenly put in some energy to either fight something or run away from something. And even though most of us are not in that kind of situation anymore, our bodies can turn this fight or flight response on with anything that our subconscious interprets, our unconscious thinks is a threat to us. So back to these tumors. So a pheochromocytoma is when you develop a tumor in the adrenal gland, and a paraganglioma is when it's in the nervous system. They're rare tumors, so they occur in 6 per million people a year. So about 30 New Zealanders a year are diagnosed with one of these. They're inherited in 1 out of 3 people. So one out of 3 of those people who were diagnosed with a tumor, one of them will be carrying a gene that's predisposed them to it. And people who have the pheochromocytomas from the adrenal glands, 10% of those will spread around the body, and 30% of the paragangliomas. So what that means to be metastatic is that the tumor has spread. The curative treatment for pheos and paragangliomas is surgery. That's the only cure for these. So we want to pick these up early when they're still able to be treated with surgery. If they have moved around the body, then we do have some treatments. We have chemotherapy, which you can see as tablets there. That's because the chemotherapy that we use is given as tablets. There's radiotherapy, traditional radiotherapy, which is called external beam radiotherapy, where a beam of radiation is shot into the body targeting the tumor. And then we have peptide receptor radionuclide therapy. And for Pheochromocytomas and Paragangliomas, we have two different types. So in these neuroendocrine cancer forums, a lot of discussion about lutetium dotatate PRRT, or often that's just referred to as PRRT because that's the type of PRRT that people with pancreas and small intestine nets get. But for pheos and paragangliomas, you can also have a different type of targeted radionuclide therapy, which is called MIBG, and that just uses a different transport system to get into the tumor cell. I've put a line in here. There are some targeted medications if you've got particular types of tumors. Those are not funded in New Zealand, but these treatments over here, the chemotherapy, the external beam, and the PRRT, are more effective treatments anyway. So we have the most effective treatments available in New Zealand, but there are a couple of other treatment types that if you're looking up online, you might come across some different types. So just a little bit more about inherited pheos and paragangliomas. As I mentioned, this happens in one of 3 people who are diagnosed with a pheo or a paraganglioma will be carrying a gene that's predisposed them to having this tumor. What that means is that other members of their family might also have that gene, so other members of the family should be tested as well. It also means that we can do a gene test to find out what gene problem they had. And these kind of fall into 2 groups. And I've put the names here, but this is not for you to remember, but just so that you know that there's 2 groups. And I've put some of the names of the specific genes just in case people here had them. I've kind of given this the name as growth factor pathways. It's not really scientifically accurate name for these, but these are in pathways that we do find in other cancer types. They tend to be really slow growing and can usually be completely treated with surgery. So for most of the people who have these types, the surgical treatment is enough for them. And then there's this other type which relates to cell metabolism, which I'm going to explain a little bit more about what does it mean, cell metabolism. And this type, they really only happen in pheos and paragangliomas. There's always exceptions. Nothing is 100% when it comes to the human body, but these are very much related to Pheochromocytomas and Paragangliomas. And these ones do have a tendency to spread around the body. And I've put SDHB first because this is a gene change that's most common in New Zealand. So in our context, we come across people who are carrying SDHB more often than the other genes. But we do know that there are some people with the other genes as well. So I just want to mention those 2. So what does cell metabolism mean? Well, cell metabolism is how our body changes food, the food that we eat, into the energy required for us to do every single thing that we do. Every single thing that our body does requires energy. So every heartbeat, every breath that we take, every movement that we make, every gurgle of our stomach, all of that requires energy. And this complicated looking cycle is our body's preferred way to make energy. So this is not the only way that our body can make energy, but this is the chemical reaction that our body prefers to use. And in the cell metabolism problems that some people carry in their DNA, it blocks this metabolism from working properly. So the body's not able to generate within the tumor cell. The tumor cell's not able to develop energy properly. So what does this mean? Well, our body obviously needs to have energy to do everything. We know that when these genes get damaged and this reaction is not working properly, that we get tumors. And we understand that to a certain extent. We understand that once the cells get under stress, they produce all of these factors that make the tumor grow. But what we don't understand is, how do these survive when such an important function is lost? So I just want to, before Dan talks about his research, I just want to briefly talk about the research that that I'm doing. And I'm just showing this picture to show that types of results that I'll hopefully be getting soon, where we're looking at all those chemicals to see how are those chemicals building up in the cell and how is that cell surviving, even though it's lost such an important function. You definitely don't have to remember any of this, but it's just to try and give an idea of what is the purpose of the research that I'm doing, is to try and work out how do these cells stay alive despite not being able to use energy properly. So that was all that I was going to talk about. Before I move on, are there any questions? So the most common sign is high blood pressure. So if a person has high blood pressure and it's really difficult to control, then that is someone who should get investigated for one of these, along with some other causes of high blood pressure. The test, so testing the adrenaline and the noradrenaline levels, can be done by the GP. If the GP gets an abnormal adrenaline and noradrenaline result, they should immediately refer to an endocrinologist. And while I think everybody's experiencing a lot of issues with the wait list when it comes to the hospital, a person who has high levels of adrenaline or noradrenaline would get a very urgent appointment. So I can only speak for my department, but I think all departments across the country would aim to do this as well. So we would aim to have someone with high levels of noradrenaline or adrenaline seen within 2 weeks. So that's quite unusual for most conditions in our hospital system at the moment, but that is what we do in our department, and most departments would get people with this condition in very urgently. So we would normally not wait for a clinic, we would get them to just come into hospital. So that depends where the tumor is. So if the tumor is in the adrenal gland, it means removing one of the adrenal glands. But these paragangliomas can be anywhere from the neck to the bladder. So it will depend where in the body that surgery is. So if it's in the neck, then you'll usually need you'll have a head and neck surgeon. If it's down in the abdomen, then you would have a surgeon who usually operated on the abdomen, and they can because they can occur anywhere from here up in the neck to down here in the bladder. It's very dependent on where your tumor is, what your surgery would be.
I am a medicinal chemist is my job title, which means I as you said, am a scientist who is involved in the discovery of new medicines, new ways to discover medicines, and new technologies to discover medicines. And I'm a senior research fellow, and I'm based at the Auckland Cancer Society Research Centre at University of Auckland. Veronica has very nicely set up all the explanations around the biology of these tumours. And so I'm going to build on that and talk about some new ideas that we have for therapies to target SDHB altered PPGL neuroendocrine tumours. So you may or may not be aware that people who, sorry, I'll just get my pointer out. People who get this disease they carry a copy of SDHB in their genome that's wrong. It's basically got a mutation, and that's not necessarily a problem initially, but when the cancer arises, you lose this what we call wild type, meaning the normal protein, and all you're left with is the mutant version. That's what causes the disease. So as Veronica mentioned, this protein, SDHB, it functions in the Krebs cycle, which is a complicated biochemical pathway that's involved in energy production in our cells. And more specifically, it's involved in converting a metabolite called succinate into fumarate. And our normal wild type SDHB does this really well, whereas the mutant version of it sort of does it. It doesn't completely not do it. It doesn't do it well. It sort of does it. And that's going to be quite important for what I'm going to talk about later. So as I mentioned, people will, when they have tumor initiation, it will actually have some cells that lose the good copy of this gene. And so all they're left with is the mutant. Now, what happens to this mutant protein in PPGL? So the cell actually recognizes it as a partially functional protein. And then what it does is recruits its own machinery to get rid of this protein. And it does this through a process called ubiquitination, which is basically the addition of these UB modifications on the protein. And the best way I can describe this is that it's like putting your rubbish bin out for collection, and to ensure that it gets collected, you put a bin tag on it. Otherwise, if you don't put that bin tag on there, it doesn't get collected. You've not paid for your collection. So the cell has a very similar system where proteins are tagged for disposal, and that's through these ubiquitin tags. And so the cell will degrade that protein into smaller bits so that it can be recycled into other proteins. And that's kind of like your recycling truck and coming in and taking away your rubbish. And this, as far as we understand it, is what is driving the cancer, the lack of stability of this mutant protein, and then the lack of its activity drives the hallmarks of cancer that we see in paras and pheos. So before I move any further into what we're doing, it's important to understand how medicines typically work. So Veronica actually mentioned that there are some targeted treatments for PPGL. And what she means by that is medicines that specifically target a gene or protein in the cell that's predicted to be driving the cancer. And so medicines, particularly in anti-cancer targeted treatments, are usually blockers of function. And I'll just explain a little bit what that means. So you've got a protein, and their function is involved in either a normal function or something that's driving the cancer. And basically we have a natural substrate that comes and binds to that, and then that gives you function. that basically activates the function. And this mechanism is very similar to how you open a locked door, where the natural substrate is a key. The door has a very specific lock, and the very specific key unlocks the door, and then that's how you have the function, which in this case, I guess, is opening the door. Now, how do blockers of that work? So a drug basically is it's like another key, but it's broken off at the end. So it will go in and get stuck in the lock. And then you can no longer put your normal key in there because you've got a stuck broken key in there, and you can't open the door. And so this is basically an analogy of how a lot of drugs work in cancer therapy. They target a very specific protein that's either overexpressed or overactive in the cancer relative to healthy cells, and will design and synthesize and test a very selective molecule that blocks that interaction that the cancer cells are dependent on for growth. So if we move back to SDHB now, the point I want to make here is that because this is not an overactive protein, it's actually the opposite. It's the fact that it gets rapidly degraded and inactivated is what drives the cancer. So our standard approach of using a blocker is not going to work here. So we have to think of something different. So the question is, can we, using some medicine, activate SDHB? And the idea is essentially to develop new molecules that are called deubiquitinase targeting chimeras. Now, what do I mean by that? So the term chimera actually comes from Greek mythology, and it means two heads. And that's essentially what these molecules are. They have two heads or two separate ends with different functions. There's a molecule that binds to SDHB on one end, and then there's a molecule that binds to an enzyme called DUB. Now, why would you want to do that? So I've explained how the problem here is the addition of these ubiquitin tags, which act as bin tags for destruction of this protein. What this DUB enzyme does is cleaves these tags off. And what that results in is a removal of the tags from the protein and a blocking of its degradation in cells, so that we stabilize and rescue the expression and function of the protein in the cell, even though it's mutant. So what does that mean? So before I explained how the mutant version of the protein, which is driving the cancer, is actually only partially functional. It's not fully functional or non-functional. But by targeting the protein in this way to stabilize it, we massively increase its expression and activity so that it's akin to a smaller amount of wild type or normal SDHB protein. So in this way, we can kind of rescue the normal function of the protein in the cell and hopefully reverse the disease. So more specifically, what are we actually doing? So we have recently received some funding from the SDHB Pheopara Coalition, which is a consortium based in the US, and it's funded by philanthropy and various industry partners to fund research into the biological understanding of SDHB mutated neuroendocrine tumors, but also to develop novel therapies for these cancers. And what we're proposing to do as part of this work is obtain from the National Institute of Health the best cell-based in vitro models. And by that, I mean models that we can grow in the lab that have been only very slightly modified from patient samples, And then we're going to implement some sophisticated genetic engineering techniques to control the levels of normal SDHB and the mutants that are found in patients where they have aggressive metastatic PPGL. And then we're going to look at downstream pathways of SDHB and how changing the levels of mutant SDHB actually affects cancer growth and various cancer hallmarks like migration and invasion. Alongside that, we would like to then also look at some novel chemistry to discover molecules that bind selectively to SDHB and its mutants. And then ultimately, we would bring objectives 1 and 2 together to design these chimeric molecules that have the 2 parts and actually test the idea of it being a medicine that brings into proximity the stub enzyme so that it can cleave the bin tags off and stabilize the target protein. So that's all I have to say. And these are the people that are involved in the project. Obviously, would like to thank Michelle for inviting me to speak at this evening, but also Veronica for all the input and collaboration with the project thus far. And thank you for listening.